Assalamualaikum warahmatullahi wabarakatuh. Hi everyone, my name is Sakia Maharani Dewi from Biology Education 2019 Universitas Negeri Semarang. Today I'm going to talk about autoclave function in the sterilization process of tools and materials in microbiology experiment. This presentation will be in 3 parts. First, I'm going to talk about introduction of my presentation. Second, I'm going to talk about methods, and third, I'm going to talk about result and conclusion. First, I would like to talk about introduction. What do we need to know? Sterilization is the process of removing all microorganisms, including the spores, contained in an object by destroying it through high-pressure temperatures. So, sterilization is one of the basic techniques in tissue cultures laboratories to prepare samples, tools, or materials used to be sterile or free of contaminants. With sterilization, contamination can be avoided, be it contamination of biological agents, chemicals, and others. Contamination can lead to false positives and negatives, which can render research results pointless and the spread of harmful biological agents such as pathogenic microorganisms that can harm laboratory workers. Then, sterilization is divided into 2 types, namely wet sterilization using autoclave and dry sterilization using oven for equipment that resistant to high temperatures. Wet sterilization is a method of sterilizing media and tools by utilizing the result of evaporation of water, where water steam is produced. Wet sterilization method uses an autoclave at 121 degrees centigrade for 15 minutes. Okay, now I would like to move on to methods. First thing to do is prepare the tools and the material or medium that will be sterilized. Second, cover the tool with a cotton or cotton plug. Third, enter the tools that will be sterilized into a plastic and tie it tightly with rubber. Then, prepare the autoclave and fill it with water to the limit. Put all the tools and materials to be sterilized. Put it on a stove and wait the thermometer showed until 100 degrees centigrade for about 10 minutes. When the manometer displayed a pressure of 2 atmosphere and the temperature of 121 degrees centigrade, hold it for 15 to 20 minutes. When it's finished, turn off the stove and open the lid and wait. For the saline medium, after being removed from the autoclave, immediately put it on a horizontal place and tilt it at an 45 degrees. Now we talk about the result. Autoclave would easily remove the bacteria using saturated steam at 121 degrees centigrade for 15 to 20 minutes. The purpose of heating is to destroy pathogenic bacteria and harmful spores. Second, before sterilized the glassware using autoclave, all tools are covered with paper or can be coated with aluminum foil. The purpose is that all tools that are sterilized are completely sterile from microbes. So, some of the main things that must be considered when carried out with sterilization are: first, sterilization depends on steam, Therefore, the air must be completely emptied from the sterilizer. Second, all tools are sterilized must be exposed to steam. Therefore, the tools must be placed in a standing position. Third, the measured temperature reached 121 degrees centigrade and was maintained that high for 15 to 20 minutes. Lastly, I would like to conclude today's presentation by summarizing the information. So, the function of sterilization is to remove all microorganisms that are on or in an object in the microbiology experimentals. Physical sterilization can be done using hot air or steam with high pressure with the steam temperature of 121 degrees centigrade for 15 to 20 minutes. Okay, I think it's enough. That's all of my presentation about sterilization. I hope this can be useful for you. Thank you for watching. Assalamualaikum warahmatullahi wabarakatuh.